Hi everyone, welcome back to Microbiogenics. Today I am going to discuss about the two pathways of apoptosis. So apoptosis is basically divided into two pathways, intrinsic pathway and extrinsic pathway. So what is the difference between these two pathways? In the intrinsic pathway, the target cell that is to be killed, the apoptosis is initiated within the cell. The target cell itself gives rise to the apoptotic pathway. That pathway that is initiated by the target cell itself is called the intrinsic pathway. So, it is initiated by the target cell. In the intrinsic pathway, the target cell itself gives rise to apoptotic pathway. So, if this is the target cell, so apoptosis will be initiated by this cell. So in the intrinsic pathway, the target cell itself gives rise to the apoptotic pathway. So this type of pathway is called intrinsic pathway. Extrinsic pathway is that when the target cell is not able to initiate apoptosis and it requires the help of other cell. So if this is the target cell, it requires the help of other cell for being killed. So what happens? This cell will bind with another cell. This cell will bind with another cell and this cell will help in the killing process of this cell. This cell will help in the killing of this cell. So this type of pathway is called extrinsic pathway. Another difference between the intrinsic and the extrinsic pathway is that intrinsic pathway of apoptosis requires the involvement of mitochondria. So here mitochondria is involved. But in the extrinsic pathway, mitochondria is not involved. So these are the two basic differences. These are the two basic differences between the intrinsic and the extrinsic pathway. Now the question is that how the body detects the abnormal cell or the target cell and how it is differentiated from the normal cell. Why normal cells are not killed by apoptosis? There must be a marker of apoptosis. So what is the marker? The marker of apoptosis is a lipid that is present in the plasma membrane. So what is the marker? The name of the marker is phosphatidylserine. This phosphatidylserine differentiates a normal cell from an apoptotic cell. So what happens? This phosphatidylserine is generally present in the inner leaflet of plasma membrane. So cells have plasma membrane and plasma membranes have two leaflets. One is the outer leaflet and another is inner leaflet. Plasma membrane is a double layered membrane. The outer layer is called the outer leaflet and the inner layer is called inner leaflet. So this phosphatidylserine is generally present in the inner leaflet of plasma membrane. In normal cells, phosphatidylserine is normally present in the inner leaflet of plasma membrane. But in the abnormal cells that is to be killed by apoptosis, that type of cell expresses phosphatidylserine in the outer leaflet of the plasma membrane. So this phosphatidylserine will be present outside the plasma membrane. So it will be flanked outside. Phosphatidylserine will be flanked outside the cell by the target cell or the abnormal cell. So this phosphatidylserine acts as a marker of apoptosis. So by seeing this phosphatidylserine, this abnormality is detected by the body and then the apoptotic pathway initiates because this abnormality is recognized by the body. So this is how the normal cell is differentiated from the abnormal cell and the abnormal cell is killed by apoptosis. Now we are moving into the intracellular pathway of apoptosis. So as I said that the intrinsic pathway is initiated by the target cell itself. Target cell means the abnormal cell. The target cell might be a diseased cell or there may be some injury in the cell or there might be oxygen and nutrient limitation within the cell. So these types of abnormalities give rise to the intrinsic pathway. So when these type of abnormalities are present in the target cell, the intracellular pathway of apoptosis initiates. Once the intrinsic pathway is activated, various types of apoptotic proteins are activated. And as a result of these apoptotic proteins, an enzyme is released from the mitochondria. So as I said that mitochondria is involved in the intrinsic pathway. So how is the mitochondria involved? Once the intrinsic pathway is activated, the apoptotic proteins causes release of a mitochondrial enzymes from the mitochondrial intermembrane space to the cytosol of the cell. Mitochondria is a double-layered organelle. It contains an inner membrane, it contains an outer membrane and an inner membrane. And the space between these two membranes is the intermembrane space. So this is the intermembrane space. So what the apoptotic proteins does is that this apoptotic proteins causes the release of a mitochondrial enzyme that normally resides within this intermembrane space. The name of the enzyme is cytochrome C. Cytochrome C is a well-known enzyme and it has role in photosynthesis, and this cytochrome C is present within the intermembrane space of mitochondria. So as a result of the apoptotic protein, as a result of the action of apoptotic proteins, the cytochrome C protein is released from the intermembrane space into the cytosol of the target cell. And once the cytochrome C is released into the cytosol, this protein interacts with other proteins. The name of the protein is APAF proteins. What will the cytochrome C do? Cytochrome C will go and bind with the APAF proteins. And what will happen next? Once the cytochrome C binds to the APAF proteins, these APAF proteins will oligomerize. Oligomerization means more than one APAF proteins will come and bind with each other. So there will be oligomerization of APAF proteins will occur, and this will give rise to apoptosome formation. The end result of oligomerization of APAF proteins is the formation of apoptosome. As a result, apoptosome will be formed. Apoptosome will be formed, and once the apoptosome is formed, procaspases will be converted into activated caspases. So initiator procaspase will be converted into activator caspases, and the other effector caspases will be activated. So a proteolytic cascade will be turned on, and the net result is the target cell death. So this is how the intrinsic pathway of apoptosis is carried out.